This is part 4 of the cell structure and function lecture. We left off with this slide, which is a summary of transport processes. There's 2 parts to this slide, and we didn't get very far on it, so I'm just going to start over. All right, so the different processes are listed on the left, starting with diffusion. The description is that diffusion is a passive process. Solutes move down their concentration gradient from where their concentration is high to where their concentration is low. The factors that affect the rate of movement is the steepness of the concentration gradient. Now, what does that mean? That means if, for example, here's your cell, and inside the cell we know is 0.85% saline or sodium chloride. And let's say that outside the cell is 1%. All right, the concentration gradient is the difference between the 1% and the 0.85%, which is just 0.15%. But here is one where the concentration gradient would be much steeper. Let's say that you've got your 0.85% in your cell, and outside is a solution that is 10% saline. That is a much steeper concentration gradient, and it's going to have a much faster rate of diffusion into the cell. The size of the molecules. Small molecules diffuse faster. Electric charge. The electric charge across the membrane. If there's a negative charge inside the membrane and the charge of the particles is positive, then they're going to be attracted to the negative interior of the membrane. Lipid solubility. So how soluble is that substance in the phospholipid bilayer? The more lipid soluble, the faster the diffusion. And we talked about substances that are soluble in lipids are fatty acids, oxygen, carbon dioxide, and just small fats or oils, and alcohol. Alcohol is lipid soluble. Temperature. The higher the temperature, the faster the rate of diffusion. And then channel proteins. The more channel proteins that you have for a substance, the faster the rate of diffusion. So osmosis is just the diffusion of water, and that's the one that is dependent on the concentration of the solution inside the cell versus outside the cell. Wherever you have the highest concentration of solute, is where water will move. So water will move toward the higher solute concentration. Okay, this facilitated diffusion is another passive process, but it requires carrier proteins. Active transport is an active process. It requires energy, a carrier protein, and it also moves substances against their concentration gradient. So instead of moving from high to low, they're moving from low to high concentration. All right, then there's vesicular transport, and we've got endocytosis. There were 3 types of endocytosis. There was pinocytosis, phagocytosis, and receptor-mediated endocytosis. And all of those are ways to bring material into the cell through a vesicle. And exocytosis is exactly the opposite. These vesicular transport does require ATP, as you can see in the table. Okay, let's look at number 12 and 13. During digestion, the concentration of hydrogen ions in the stomach rises to many times that within the cells lining the stomach, where hydrogen ions are produced. Is this type of transport process involved passive or active? Well, if the cells that are producing the hydrogen ions keep producing hydrogen ions, and releasing hydrogen ions into the stomach, then the hydrogen ions are moving from their low concentration to their high concentration. So this has got to be an active process. And by the way, what's happening here is the buildup of hydrogen ions. Remember back to the chemistry chapter? The buildup of the hydrogen ions is lowering the pH and making the stomach more acidic. Number 13, when certain types of white blood cells encounter bacteria, they are able to engulf them and bring them into the cell. What is this process called? You may remember it's a type of endocytosis called phagocytosis. Okay, so the cytoplasm of the cell is the topic of this page. The cytoplasm is anything between the plasma membrane and the nuclear membrane. And the cytosol is the fluid, and then the organelles are the structures that are found within the cytosol. Cytosol contains dissolved nutrients, ions, proteins, and waste. It differs from the extracellular fluid because of the sodium-potassium pump. The cytosol is going to contain more potassium than the extracellular fluid and less sodium than the extracellular fluid. And the cytosol also contains higher concentrations of proteins. And those proteins have a negative charge. So they contribute to the negative charge inside the plasma membrane. And they also contribute to giving the cytosol a thicker consistency, a more viscous or more denser consistency. And there may also be inclusions inside of the cytosol, which is materials that are insoluble. The organelles are the structures that reside in the fluid within the cell. They have different functions. The cytoskeleton are protein fibers. They consist of microfilaments, which are the smallest, intermediate filaments, and then microtubules, which are the thickest. This image just shows you some microfilaments and what their role is. Okay, microfilaments are the thinnest, and they help provide attachment of certain structures to other structures, but they also are found within microvilli, which are the structures that are they're foldings of the plasma membrane that increase the surface area. So microfilaments are the thinnest. The intermediate filaments are the purple ones, and they sort of provide a framework inside the cell. And then you can see a microtubule, which are thicker. Microtubules help during cell division. Microfilaments are the thinnest. They are usually composed of a protein called actin. They attach the plasma membrane to the cytoplasm, and they participate in muscle contraction. Intermediate filaments are medium size or intermediate in size. They strengthen and stabilize the cell. And microtubules are the largest, and they are built from the protein tubulin. They give the cell strength. They anchor organelles in place so they're not moving around everywhere. And they also form the spindle apparatus during cell division. Oftentimes when I'm teaching biology, I refer to the microtubules as spindle fibers during cell division. Microvilli are areas of the plasma membrane that are they're finger-shaped. Anyway, they're foldings of the plasma membrane. And what they do, they have microfilaments inside them, but their purpose is to increase the surface of the membrane. Cells like the cells lining your intestines, which are absorbing nutrients from the food that you digest, are able to absorb a lot more nutrients with these microvilli. Centrioles are involved in cell division, and they are composed of microtubules, and the pattern is there are 9 microtubule triplets. So each group of microtubules has 3 in it. So each one of these right here has got 3 microtubules in that group. These centrioles produce the spindle fibers, which are microtubules that help move the chromosomes when the cell divides. Cilia are long hairs, and there's many of them covering the surface of a cell. All cells do not have cilia, but the ones that do use the cilia to move materials over the cell surface. For example, our respiratory tract, the cells that line our respiratory tract are lined with cilia to help move mucus and other harmful substances that get trapped in the mucus out of our respiratory tract, so that we don't get lung infections. There's another function of cilia in males. They have cilia in certain cells of their testes, which help move the sperm along until they are ready to move on their own, until they're mature. So cilia are little hairs that move substances across the surface of a cell. And a flagella, speaking of sperm, the only cell in the human body that has a flagella is in males, and it is the sperm. It's a tail-like structure that functions in movement. Ribosomes are organelles, but they are not membrane-bound organelles. So every type of cell has ribosomes. Ribosomes can be free in the cell, or they can be attached or fixed to the endoplasmic reticulum, to the rough endoplasmic reticulum. We call that the rough ER. And their function is to make proteins. They function in protein synthesis. Proteasomes are organelles that break down proteins. So they contain enzymes called proteases. They are just vesicles that contain these proteases, and their function is to break down proteins. The rough endoplasmic reticulum functions in modifying, packaging, and synthesizing proteins. So the primary product of the rough ER are proteins, and the primary product of the smooth ER is lipids and carbohydrates. So 4 major functions of the endoplasmic reticulum is the synthesis of substances. The rough ER synthesizes proteins, and the smooth ER synthesizes carbohydrates and lipids. The storage of materials, isolating them from the cytosol. For example, the smooth endoplasmic reticulum in your muscle cells is actually called sarcoplasm. No, it's not. It's called, I can't think of it. It's the sarcoplasmic reticulum. That's what it is. Anyway, it's the smooth ER of muscle cells, and the smooth ER stores calcium ions, lots of calcium ions, which are released prior to a muscle contraction. So ER can also function in storage. It can function in transport of materials through the cell because it's a network of channels. And the smooth ER especially can help with the detoxification of drugs or toxins. So you would find a lot of smooth ER in the liver. All right, and then here you have the smooth endoplasmic reticulum does not have ribosomes. It synthesizes lipids and carbohydrates such as phospholipids and cholesterol, steroid hormones like testosterone and estrogen, triglycerides and glycogen, which is found in skeletal muscle and liver cells. That's what is produced when our skeletal muscle cells and our liver cells remove glucose from our blood and lower our blood sugar. The rough endoplasmic reticulum has ribosomes, and so it's mainly functioning in the synthesis of proteins and then packaging them up and delivering them to the Golgi apparatus.